multiple departments at Stanford, and my background is actually in math, and then I got a PhD in statistics without any biology. But after, kind of functioning as a statistician for quite a while, I realized that the biological universe was incredibly fascinating to me, and I really wanted to make a career transition, which I did about 15 years ago. I was really especially motivated by two biological problems, which I really at the time only understood from a lay perspective, and they were ones that I'm sure you are all aware of and very interested in as well. One of them being cancer and the other being viral infections. And so I have, when I actually started work as a computational biologist without really knowing anything, I worked in the field of cancer genomics right at the time that sequencing was beginning to be available. And one of the projects that I worked on was a project around trying to discover gene fusions in cancer. And this was suggested by my mentor, Pat Brown. And the reason that he suggested it was that one of the defining features of cancer genomes is actually their incredible genomic instability at the level of chromosomes. And you can see that on the left here with an incredibly aneuploid chromosome cytology from a tumor cell of a glioblastoma. And this is very typical of cancer cells, okay? They don't have normal chromosomal configurations. Though still, a lot of focus is on studying cancer at the point of, at the level of point mutations, where we try to find individual single nucleotide variation, okay? One of the fascinating outcomes of the sequencing revolution has been that we've actually found that normal epithelial and normal cells in your body have the identical mutations in driver genes that cancer cells do. Okay, so there's a real suggestion that genomic rearrangements define and drive cancer. Another huge kind of, biological problem, which I know that everyone is also familiar with now, especially in the era of SARS-CoV-2, is that viruses are an incredible threat to human health. Today, because of vaccines, we're at a moment in history where we have an approximate handle on being able to control them. Throughout history, they've been incredibly deadly, okay? So these are two examples of biological problems that genomics really wants to tackle and have incredible impact. Okay, so is there a common theme in terms of what our field as computational biologists could contribute to understanding cancer and understanding viral biology and evolution? Okay, and so what I want to really tell you about for the rest of the talk is a different way of thinking about these problems than the one that we typically, the approach that we typically take as a field. Okay, so today we have better and better compute, we have more and more storage, and we are still as a field generally trying to keep pace with the reference genomes of cancers and viruses among other species that we attempt to sequence and understand. Okay, and we are still building knowledge towards the first incredible advance in computational biology, which was the sequencing of the human genome more than 20 years ago. We now appreciate that it is a sequence of a single human genome. And the more and more we attempt to expand the knowledge of the human genome to diverse populations, the more we understand that it is necessarily incomplete. Okay, this is a quick example from a recent study where there were just about 100 or so African individuals that were sequenced, and they found more than 300 megabases of missing DNA and genes. Okay, in cancers, this is especially, the incompleteness of the reference genome is especially apparent. The kind of crazy chromosomal configurations that I showed you in one of the earliest slides, we now understand some of the mechanisms that underlie this, and it's called chromothripsis. Literally what happens is DNA can get shattered and rearranged, almost like a combinatorial rearrangement of puzzle pieces. You can see that the resulting chromosomes really have no relationship to the human reference genome. Beyond cancers, the scale of the living world means that it is really basically impossible to store reference genomes. There are more than 10 to the 30th bacteria on Earth, more viruses. In the plant kingdom, there is an incredible amount of structural variation. And although we still attempt to assemble reference genomes, they're almost certainly incomplete. Okay, so I think these are strong biological reasons to try to avoid references, but are there also statistical ones? And I would say, as a statistician, yes. Even if you had a perfect reference genome, we know that reference genomes are littered with repetitive elements. And because of that, alignment algorithms they become complicated multi-step procedures. This is an example of a workflow for a typical alignment procedure. They're very complicated. They have to rely on sometimes randomized heuristics in order to make the search efficient. And for all of these reasons, it is difficult or impossible to obtain valid p-values at the end of taking data that has been run through an aligner. Okay, so I'd like to just ask, why do we use references? I mean, alignments are useful, but why are we actually using them? Okay, and I would say one of the major reasons that we use reference genomes is as a guidepost to the fundamental scientific step, which is inference of differences between conditions. So, for example, in cancer, we might want to align to a reference genome so that we can then perform statistical inference on what is different about the cancer versus the normal. Or in a microbiome, we might want to perform inference on what is different after you have been treated with antibiotics or not. In the case of plant genomics, we might want to perform inference on whether this crop variety is better resistant to drought or makes better fruit. Okay, so that's a really powerful kind of reference genomes enable this kind of scientific inference, which is great, but they have all these limitations that I started the talk with. So the question is, could we bypass reference genomes and still provide and obtain the statistical inference that we eventually want to have, which is, fundamental. Okay, so for the rest of the talk, what I'll tell you about is our work, which I think is a very strong first step in this direction. And it derives from formulating a probabilistic model directly so that we can perform statistical inference on the data that we observe from sequencing, K-mers or FASTQ files, without going through this intermediate process of alignment, okay? And in order to give some intuition for what we're gonna do, I'd like you to consider that there are a variety of different biological problems, from cancer genomics to viral variants that are emerging, to mobilization of DNA or infection of phages into bacteria that have a shared biological feature. And that feature is also shared by RNA splicing. And that's an example that I'll talk about in a little bit. That feature is that sequences in these, when we are interested in the differential conditions between, for example, a phage integrating into a bacteria versus not, there's a shared biological feature, which I will call sequence diversification. And that is the local sequence context in the genome with and without the phage is different. So if you see a sequence here and you have the phage, the sequence near it is going to be different than the sequence without it. Okay. So there are many, many examples, which I will turn to later, that fit into this probabilistic formulation, from microbial evolution to spatial genomics, cancer genomics, and more. Okay. So the current approaches to attacking the problems that I described to you in the previous slide all follow essentially the same workflow of taking FASTQ files, aligning them with throwing out unaligned reads that don't match the genome, and then performing some kind of statistical inference. And they have shortcomings, which include they're being fragmented in that you have to pre-specify a specific pipeline and algorithm to run. They're time consuming. We know that alignment is computationally intensive, and they have unknown sources of errors. Okay, so I'm now going to tell you about NOMAD, which is a reference-free and sensitive approach to direct statistical inference on k-mers. And this is the work of two tremendously talented people. It's really a team, an incredible team effort, Caitlin Chang and Tabor Baharov. I think they might both be here, as well as Vanya Zeludev. Okay, so like I said, we are going to be really treating genomic inference as a direct statistical procedure on sequencing data, raw sequencing data, and bypassing alignment completely. So while I could talk about the math later, and I would love to, I'm pretty much going to just share this high-level view of what I mean by sequence diversification. So consider a problem like RNA splicing, which falls into this category of attempting to do inference directly on sequencing, raw sequencing reads. The question is, we have two samples. Sample 1 might be cell type 1 and sample 2 cell type 2. Sample 1 predominantly splices exon 1 to exon 3, whereas sample 2 to exon 2. Okay, so I've given an example. Oh, sorry. Okay, so sample 1 and 2 are obviously switched at the top here. Sorry. But I've given you an example of the configuration of the exon splicing at the bottom and an example sequence that might be underlying this toy kind of box plot. So what is the kind of statistical or probabilistic model underlying this difference? It is that if we observe a sequence S at the beginning of this read or RNA transcript, the probability of seeing T, another subsequence, some fixed distance downstream of S, is a function of the sample identity. Okay, so it is sample specific, this probability distribution. And that's really the key kind of high level idea that we're going to be using to work with. Okay, so what do we do in this algorithm NOMAD? Okay, we are going to be processing raw sequencing reads. And you can think of it in the simplest case. Of course, we are scaling this, as I'll tell you about. But just in the case of 2 reads, we get a single k-mer or sequence from sample 1, another sequence from sample 2. We catalog their k-mer compositions in the following way. We look for the first part of the sequence in sample 1. It's going to be AAA in this toy example in these 2 samples. And we then build a contingency table. Very simple, basic statistical object, which has 2 different subsequences, target 1 and target 2. Because these subsequences are the ones that follow L nucleotides away from that first subsequence. Then we do this for each sample. We catalog the counts, and we perform some statistical procedures that provide closed form valid p-values without any kind of resampling needed. And this is very important because it allows this algorithm to be incredibly efficient. Okay, so compared to existing methods, what NOMAD's workflow is, it takes in the raw FASTQ files, it sorts them and kind of parses them into contingency tables. It performs valid statistical inference incredibly fast and incredibly quickly, and then it does post facto inference or for interpretation. So sorry, when I mean inference, I mean human kind of interpretation. And it has allowed us to, in the same algorithm as I'll tell you about now, detect alternative splice variants more powerfully than state of the art. VDJ recombination, which is the basis of how antibodies and T cells are generated, viral strain variation, and other examples, again, that I don't have time to tell you about. Okay, so I'm going to give a snapshot of the results. What I will tell you about derived from application of the identical algorithm in its default parameter regime. So there are, of course, lots of parameters that we can change, for example, k-mer length and other things. But everything was just run kind of in default mode. Okay, so I'll tell you a little bit about SARS-CoV-2 and human single cell RNA-seq data. I don't think I'll have time to tell you about lemurs. Okay, so SARS-CoV-2 is obviously an incredibly serious public health threat. And today, I don't know if people really remember how Omicron was first identified in South Africa. It was identified clinically. It was not first identified genomically. Clinicians realized that there was an uptick in cases, and then they went into the sequencing surveillance data that they had performed and had to kind of combine alignment-based procedures and manual curation to discover the variant. Okay, so we just grabbed the sequencing data from about 70 patients in South Africa in that first genomic study that clinicians used to characterize the Omicron variant. And we ran NOMAD, which is the algorithm that I talked to you about. We also have a control for NOMAD, which for time reasons I won't go into. And one of the really exciting things to us was that NOMAD identified k-mers that were highly enriched in being proximate to the known Omicron variant without any knowledge of the SARS-CoV-2 genome, without any knowledge of the Omicron variants. Okay, so again, we are running a statistical algorithm on raw sequencing reads from patients, and yeah, okay, 5 minutes, yeah, inferring exactly where, basically trying to infer which k-mers are sample specifically diverse. So here's an example of a sequence that NOMAD identified. After the NOMAD calls, we sought to interpret it. We aligned it to the SARS genome, and it aligns immediately upstream, as would be expected if this algorithm were performing as we wanted it to, before a set of Omicron-specific mutations. Okay, here's another way that we can run NOMAD. It doesn't require any reference genome. And so for that reason, while we can take the k-mers that our output by it and align them to reference genomes, we don't have to. We can actually go directly to an in silico translation of these k-mers to predict protein fragments that we can then align to databases. We call this in silico translation. We perform this on another data set, and I guess the slide got cut off. So by ordering the enrichment of the protein domains in NOMAD compared to other the controls, we identified that the spike protein, totally de novo, had the highest mutational burden. Okay, the last minute or two, I'm going to give you a final snapshot of NOMAD's application to human single cell biology. Okay, one of the major mysteries of the human genome, and one that has been very difficult to study with alignment-based procedures, is why there are gene duplications and whether they have functional differences. Okay, here's an example of a gene that has this feature, MyL12, which has 95% 95% nucleotide similarity between two homologs of the gene that are positioned immediately next to each other on the human genome. Alignment-based approaches simply cannot distinguish which of these two isoforms are expressed, and NOMAD is able to do this basically able to resolve cell type specific expression differences of these two isoforms without any kind of guidance or training. And I think really opens the door to a huge amount of new biology, for example, to study human immunology. I think for time reasons, I'm going to skip this, but the major histocompatibility complex is a gene locus that is one of the most important for protecting our bodies against cancer and viruses, but has large numbers of homologs that make alignment-based approaches essentially unable to resolve haplotypes efficiently. Okay, so in summary, there are really myriad applications of NOMAD analysis in the biological realm. And I think one of the most important to me is an implication for health disparities. So, you know, the human reference genome was built from a Caucasian individual. And even though we are, as a field, attempting to rectify that situation, in reality, the vast majority of sequenced genomes come from a certain subset of individuals. An advantage of going completely reference-free is that you have no bias towards finding variants that are associated with diseases based on what we know about reference genomes from, at least in the US, Caucasian individuals. We're about to, extend this algorithm to be very versatile and work with 10x genomics. It already works really well with SmartSeq2. And there are just myriad applications here to viruses, to microbes, and other applications. From the statistical side, we have one statistical approach that we published in all of this work, by the way, is published on the bioRxiv. But there are many important and open statistical problems that this work provokes, and we're very excited on working on them. And so I would like to thank everyone that has contributed to this work and the funding agencies.
Could you compare and contrast that to Callisto and other existing reference-free?
Sure. So it's completely different. So Callisto, it's a pseudo alignment, right? So it's not reference-free. It requires a reference. It requires a set of transcripts, and then you take those transcripts. You basically make a database of their k-mers, associated k-mers, and then you match k-mers in your sample to those k-mers. So there are some very strong differences between this approach and Callisto. I could list more of them if you'd like, now or later.
But I wanted to just ask a question that automatic splicing, and to which extent, you get a better sense of automatic splicing is versus ability to build the whole, model as, is the final transcript.
Yeah.
So that seems to be maybe a difficulty here.
So it's interesting. Thank you for asking that question, because my group is actually focused on alternative splicing until really I stepped back about a year ago. I would say this approach has, I would say, more power to detect alternative splicing precisely, both in its discovery power and also, I didn't have time to talk about here, but it's on the bioRxiv paper. I'd be happy to talk about it, that we are able to basically, I would say, improve and denoise the splice junctions that can be derived from any RNA-seq data. So I think that's one of the really exciting things to me, is that actually, I think will improve the power of alternative splicing detection in single cell or any.
But you still will have to have an alignment to build the actual.
True, okay, and so that's a critical point that I skipped for time. For those kinds of alignments, we both do, this algorithm denoises and reduces the number of required reads by 3 orders of magnitude.
Okay, thanks.